Right. So I wanted to go over the rest of this section. In the last section, I talked about the operon being an example of how prokaryotes control gene expression. And just to review that, so an operon is made of 4 parts. We have the regulator gene, which codes for the repressor protein, which binds to the operator and blocks RNA polymerase from sitting on the promoter to transcribe the structural genes. This is an example of what's called repression. So the specific operon that we looked at was the lac operon that makes the lactose enzymes. So when lactose is not present, you don't need the enzymes to break down lactose. So the operator is blocked by the repressor protein, so RNA polymerase cannot go past it. During activation, the lactose is going to bind to the operator and pull it off. That allows RNA polymerase to bind to the promoter and then transcribe the structural genes. So then what's going to be transcribed are lactose enzymes that will break down the lactose. And then when there's no more lactose, then of course it all goes back to repression. Oh, no, wait. So today we're going to look at the eukaryotic gene expression, and this is going to occur in a couple different ways. First, we're going to look at the general structure of a eukaryotic chromosome or eukaryotic DNA, and also the structure of a eukaryotic cell before we look at the actual mechanism of how gene expression occurs in eukaryotes. So we know that the genomes in eukaryotes are much larger, and that DNA is going to be located on several different chromosomes. Compared to a prokaryote that has a single circular chromosome, and so for eukaryotes, only some genes are expressed, and some of them are not expressed at all, ever. So controlling gene expression in eukaryotes is going to be more complicated. So remembering the structure of eukaryotic genes, this is a substance called chromatin. Chromatin is the DNA double helix wrapped around histone proteins. Then you basically coil that up and into a larger thread. That thread is then curled up, and then that is also condensed into the structure of a chromosome. So gene expression in eukaryotes is partly related to the coiling and uncoiling of DNA in each chromosome. Before division, the DNA is going to coil into chromosomes. But after division, it's going to partially uncoil into euchromatin. Euchromatin, which you see here, is partially uncoiled DNA, which allows for some transcription. But you can see that some parts are still coiled, so those parts are never transcribed. Now, another way that we control gene expression is between the process of transcription and translation. So in prokaryotes, they don't have a nucleus, so transcription and translation both occur in the cytoplasm. But in eukaryotes, these two processes occur in different locations. Transcription occurs in the nucleus, translation occurs in the cytoplasm. This is going to allow the cells to have more control because you're going to be able to manipulate the RNA before it leaves the nucleus to go to the cytoplasm. So one way that expression is controlled is through the action of introns and exons. Now, remember that in eukaryotic cells, RNA polymerase binds to the promoter. It's the same type of area as in an operon. A promoter is just an area that is recognized by DNA or RNA polymerase as a beginning of a gene. So you can think of it that way. The promoter is the beginning of a gene. Whether it's in a eukaryote or a prokaryote. Beyond the promoter, when you get into the structural genes, there are 2 segments. You have introns, which you see here in blue, which are sections of structural genes that do not code for amino acids. And you have exons, which do code for amino acids. Oh, okay, hold on. A couple people just showed up. Okay. All right. So when RNA is first transcribed, we get something called pre-mRNA. This is because both introns and exons are transcribed. But of course, introns don't code for anything. They're nonsense. So we're going to have to splice the exons together, which means we cut out the introns and we attach all the exons together. This molecule here is called pre-mRNA because it contains both introns and exons. Now, there are complex assemblies of RNA and proteins called spliceosomes, which split pre-mRNA at each end of an intron and join the exons together. So the resulting molecule is mRNA.
As DNA is transcribed into RNA, it needs to be edited to remove non-coding regions or introns shown in green. This editing process is called splicing, which involves removing the introns, leaving only the yellow protein coding regions called exons. RNA splicing begins with assembly of helper proteins at the intron-exon borders. These splicing factors act as beacons to guide small nuclear riboproteins to form a splicing machine called the spliceosome. The animation is showing this happening in real time. The spliceosome then brings the exons on either side of the intron very close together, ready to be cut. One end of the intron is cut and folded back on itself to join and form a loop. The spliceosome then cuts the RNA to release the loop and join the two exons together. The edited RNA and intron are released and the spliceosome disassembles. This process is repeated for every intron in the RNA. Numerous spliceosomes, shown here in purple, assemble along the RNA. Each spliceosome removes one intron, releasing the loop before disassembling. In this example, 3 introns are removed from the RNA to leave the complete instructions for a protein.
So in this example, we have the exons, which are in yellow. Those contain the amino acid sequences, and we have the introns in green, which don't code for anything. So part of the way that we control expression is by being able to splice out these introns in between transcription and translation. So again, we've got transcription in the nucleus and translation in the cytoplasm. The immediate product of transcription is a molecule called pre-mRNA, which includes introns and exons. Introns do not code for anything. Exons are the information that you need to make the protein. So the introns have to be removed and the remaining exons spliced together to create mRNA. Only then is mRNA released to the cytoplasm where it can be translated into a protein. So the end result is an mRNA molecule that contains only exons, and then it will be able to leave the nucleus. The nucleotides in the introns can be used again during transcription of more pre-mRNA, because remember, both introns and exons are just sequences of nucleotide bases, so that can be used again to make more mRNA. So you might think, if introns don't code for anything, why are they there? Well, we don't quite understand the benefit of introns being there if they don't code for anything. But of course, there's ideas about why we would evolve to have these nonsense sequences in between the sequences we need to make a protein. One idea is that the pattern of coding being the exon and non-coding being the intron sequences could provide options for making different proteins. This is because if you remember back in chapter 8, when we looked at cellular reproduction, during meiosis, the chromosomes come together during crossing over, and parts of chromosomes can exchange information. This might result in some intron exon sequences being swapped between chromosomes. This results in additional genetic diversity. So it might just be a fact of like, if we have introns exons, it puts the exons further apart on the chromosome, which means there might be a higher chance of creating new proteins, which means we increase our diversity. Increasing genetic diversity is always an advantage in nature. The more different we are, which I find is actually interesting because culturally, like as a society, as a species, we like to think that we want to be like everyone else, right? That's what peer pressure is all about. It happens when you're kids, when you're teenagers, even as adults, we still feel peer pressure where you want to be just like everyone else. But biologically speaking, we are more at an advantage if we are different from each other, because when something like coronavirus comes out, it affects all of us differently. Imagine if we were all clones of each other and a virus like coronavirus makes its way into the human population, we would be extinct, right? But instead, we're entering the third year of a global pandemic. Even though we have like 6 million, 5.5 or 6 million people that died out of a population of nearly 8 billion, and I'm pretty sure by now we're at 8 billion, that's a significant difference. Scientists have now discovered RNA molecules that can act as catalysts to remove introns from mRNA itself as it forms in the nucleus. Until this discovery, it was thought that all enzymes were proteins, but now we know that there are RNA molecules that can act as enzymes as well. And I'm going to skip through this because this only works in person. Normally, I have you guys make like a secret sentence that contains only 3-letter words that make sense, like the cat ate the rat. And then in between the words, you're going to put nonsense letters. Each of these 3-letter words acts as a codon. That's why I had it 3 letters. And like I said, normally I want you to hide those words inside random letters. Then you're going to swap it with somebody else and you're going to try to decode. You're going to cut out the introns, which are the nonsense letters, and try to read the sentence, like the 3-letter words, which are the exons. But, oh well, maybe we can do that. Well, no, we're not going back, are we, until the next year? All right, moving on. So how do we control gene expression when transcription occurs? Most gene regulation in eukaryotes happens when RNA polymerase binds to a gene. This is the beginning or the onset. Onset is a good SAT word, cough, cough. It means the beginning of transcription, the onset of transcription. Eukaryotic cells also have regulatory genes, but they involve more proteins than prokaryotes. The main controlling factor for eukaryotes are these regulatory proteins called transcription factors.
By controlling which genes are active, a cell can take on special characteristics. Muscle cells and neurons have the same DNA but perform different functions because they use different sets of genes. A key challenge in genetics research is to understand how transcription factors find the correct DNA sequences to control genes. In the nucleus, DNA is condensed into a DNA-protein complex called chromatin. In places where genes are being expressed, there are often zones of naked DNA. We will now explore ways that transcription factors interact with these zones. Even among scientists, a common misperception is that transcription factors fall from the sky onto the DNA and test one spot at a time. Such a process is too inefficient. Instead, the transcription factor interacts non-specifically with the DNA backbone. This allows it to efficiently search the DNA for its binding site, a DNA sequence it recognizes.
That's the promoter.
The transcription factor slides along until it encounters its binding site. Then it changes conformation to interact specifically with nucleotides on the interior of the helix. Now the transcription factor can stay and interact with other proteins for transcription.
Oh, sorry, not the promoter.
Or it can become dislodged by many factors, which could include movement of the DNA or interaction with other proteins. Studies have suggested that transcription factors sometimes move from one part of the DNA to another. Short hops might allow them to bypass other proteins or to hop over compacted sections of DNA. When the transcription factor lands on another segment of DNA, short hops become big jumps. Although the other strand is nearby in three-dimensional space, it may be much farther away on the chromosome or on another chromosome entirely. There are 1,500 different types of transcription factors in human cells. They interact to create a complex language of gene expression.
Okay, I said the promoter was totally wrong. It's the enhancer, excuse me, because the promoter is for RNA polymerase. All right, so you saw in the video that the transcription factors are proteins that basically just kind of wind their way down the DNA molecule until they hit an area that they recognize and bind to. This is called the enhancer. So the point of a transcription factor is to help in the placement of RNA polymerase at the correct area on the promoter. Now you heard in the video, she said there's over 1,500 transcription factors. There's many different transcription factors that can influence one gene. So this is what the DNA double helix looks like under normal conditions. So you can see that there is a huge distance between the enhancer and the promoter region. The structural genes are located just after the promoter region, but the enhancer being so far away is one of the ways that we control whether or not a gene will be expressed. So this could be a distance of thousands of nucleotide bases away from the gene and its associated promoter. Now, when a transcription factor comes along and hits the enhancer, that's when it forms this kind of loop, which brings the enhancer and the promoter together, which then signals the RNA polymerase to start transcribing the gene. So in the video, you saw sometimes as it's going down the DNA, sometimes there's something blocking it, which causes it to break off, or maybe it hops over and it misses the enhancer. Therefore, these genes won't be transcribed. There's all kinds of different things that can happen. So the transcription factors that are bound to the enhancer are going to activate the transcription factors bound to the promoter on this side. Oop, it was already playing. Uh-oh. Let's go back. Okay.
Synthesis of an mRNA begins in the nucleus. There, transcription factors and coactivators bind to enhancer sequences. These factors are joined by general transcription factors, SRBs, and RNA polymerase II at the promoter to assemble a transcription initiation complex. When transcription begins, the carboxyl terminal domain of Pol II becomes heavily phosphorylated. The 5' end of the growing RNA is protected from 5' exonucleases by a cap of 7-methyl-G. A cap-binding complex and different kinds of HNRNPs bind to the RNA. The spliceosome, made up of snRNAs and proteins,
That's taking the
removes
introns
introns
away.
from the RNA. At a polyadenylation signal, a protein complex cleaves the mRNA and adds a poly-A tail on the 3' end. A poly-A binding protein and a cap binding complex protect the mRNA.
Okay, how about we go back and I'll narrate that because it's a whole lot of foreign language sounding things. All right, so we just want to look at the chromatin. That's like university level like detail. Don't care about that. I just want you to see the big picture, right? So you've got the chromatin, we've got a single strand here, and we have our enhancer region. Now in this case, they've put the promoter after the other enhancer region. It doesn't matter which side it's on. Just understand that there has to be transcription factors that are going to bind to both of these sites. When the transcription factors bind to those sites, that is when you have, I don't think they told you what that structure is, but basically it forms a loop in the DNA. That is a signal for RNA polymerase, which is over here, to bind to the promoter, which is here. And it starts to transcribe that DNA. Now, who's like blah, 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 blah. Now, here's where you've got the red area is the exon and the white area is the intron. So as it's coming out, you have enzyme actions or spliceosomes that are going to cut the intron out and splice the two exons together. Okay, that's the main idea. Okay, I'm going to stop there.